Skin cancer usually develops from excessive exposure to ultraviolet radiation and can also arise from certain genetic predispositions. Melanoma is the deadliest type of skin cancer due to its ability to quickly metastasize throughout the body. It is characterized by unregulated growth of melanocytes due to genetic mutations that are accumulated throughout a lifetime or through inherited mutations. Melanocytes are cells that give our skin its color by producing the melanin pigment to absorb harmful UV rays and protect our cells from sun damage.
Melanoma is the fourth most diagnosed cancer in Australia, with an estimate that 1 in 18 people are at risk of diagnosis by the age of 85. Melanoma diagnosis is far more prevalent in countries with a higher HCI, whilst countries with a lower HCI have increased rates of mortality from melanoma. Both Indigenous and non-Indigenous Australians' incident rates drastically increase with age, shown by the difference between the 45 to 64 age group and the 65 plus age group, with a 440% increase for Indigenous Australians and a 280% increase for non-Indigenous Australians. Whilst on the contrary, survival rates follow a negative linear response at age, decreasing as age increases. Further, Australian males have approximately 2.2 times higher incident rate due to thicker skin with less fat beneath them, making their skin more susceptible to UV radiation.
Next, we will look at the normal physiology of the cutaneous system, which is responsible for the formation and maintenance of multicellular tissues under environmental stresses such as UV radiation. There are three main types of cells within the epidermis: keratinocytes, melanocytes, and Langerhans cells. Melanocytes are located above the basement membrane, which separates the epidermis from the dermis at a ratio of 1 to 10 with surrounding keratinocytes, and are responsible for the protection from ultraviolet radiation. The tanning response of the skin is a defensive measure in which melanocytes synthesize melanin and transfer it to keratinocytes, which absorb and dissipate the UV energy. This process is regulated through the action of alpha melanocyte stimulating hormone on its receptor, the melanocortin receptor, that is responsible for melanocyte differentiation, and hence the transcription of genes underlying melanin synthesis. However, intense intermittent exposures to UV light cause genetic damage, impairs cutaneous immune function, and induces the formation of DNA-damaging reactive oxygen species.
Melanoma mostly occurs due to prolonged intense sun exposure, as UV light damages the DNA in skin cells, but there are several more suspected sources, including family history. This is associated with inheritance of tumor suppressor gene mutations. Having a personal history. People with a history of melanoma or other skin cancers, such as basal cell carcinoma, run a higher risk of developing melanoma in the future. Having high-risk personal characteristics, such as fair skin with little pigment, blue eyes, blonde hair, freckles, or having multiple or atypical moles. Being in an immunosuppressive state, as the immune system struggles to fight cancer cells. Or socioeconomic status. Low SES is often linked to more advanced melanoma at the time of detection, as well as a decreased perception of melanoma, while high SES has a greater incidence.
When skin is exposed to the sun, UV rays penetrate the dermis and over time cause DNA damage. Direct DNA damage causes the formation of cyclobutane pyrimidine dimers, which form between two adjacent thymine or cytosine bases. These dimers extinguish or alter the function of genes within these damaged regions. Proto-oncogenes such as BRAF and NRAS encode regulatory proteins and growth factors, which enable progression through the cell cycle, leading to regulated cell growth. When DNA mutations occur within a proto-oncogene sequence such as BRAF, it can alter their expression to form an oncogene. Oncogenes are permanently activated, which causes unregulated cell proliferation, leading to cancer formation. DNA mutations can also alter the function of tumor suppressor genes. Under normal conditions, tumor suppressor genes inhibit cell division and replication to prevent tumor development. In melanoma, the CDKN2A gene is inactivated by mutations, and the proteins encoded by the genes are not produced. Without these proteins, tumor suppressor genes are not formed, and therefore their inhibitory function is extinguished, causing melanocytes to undergo uncontrolled cell proliferation. Individuals with fair skin and red hair have a loss-of-function mutation in their melanocortin receptor 1 gene, causing impaired production of eumelanin, the type of melanin responsible for reducing UV-induced DNA damage. These individuals instead produce phenomelanin, which has limited UVB protection, increasing their risk of melanoma.
So coming to the signs and symptoms, we have ABCDEs of melanoma. So the first one represents the asymmetry of the size, like it's increasing in size, followed by the border, which is scattering, has started to scatter, and then comes the color, in which it's getting a little bit more pigmentation, followed by the diameter, which is increasing from 0.25 inches, and in the end, the colony has started to evolve now. So melanoma frequently has no symptoms, but the first indicator is usually a change in an existing mole or formation of a new lesion. So the melanoma can also form in places of your body that receive little or no sunlight, and such as in between your toes and your palms, could be in between the soles, scalp, or genitals, and it is more likely to form in a concealed location in those with the darker skin.